I'm Tanya Gruber, the Chief of Pediatric Hematology Oncology, Stem Cell Transplant, and Regenerative Medicine here at Stanford. And I want to thank Alice for giving me the opportunity to introduce the closing keynote speaker. It's truly an honor to introduce Dr. Handgretinger, who is a Professor Emeritus of Pediatrics. He received his medical degree from the University of Tübingen, where he completed his training in pediatrics and hematology oncology. He was recently the chairman of the Department of General Pediatrics and Hematology Oncology at the Children's University Hospital in Tübingen, and more recently moved to the Abu Dhabi Stem Cell Center, where he is the head of adoptive cell transfer section and the head of the pediatric service, the Abu Dhabi Bone Marrow Transplant Program in the UAE. Dr. Handgretinger's main research interests have been cellular immunotherapeutic approaches to the treatment of children with various forms of cancer in the context of allogeneic stem cell transplantation. He was one of the first to introduce immunotherapy with bispecific antibodies in children with relapse refractory acute lymphoblastic leukemia. And he's also involved in clinical trials with the anti-CD19 and 1922 CARs in pediatric relapse and refractory leukemia. He has authored and co-authored over 500 papers in peer-reviewed journals and contributed to various textbooks dealing with stem cell transplantation and immunotherapy. He's received many awards, most recently the 2021 Lifetime Achievement Award from the Pediatric Transplantation and Cellular Therapy Consortium. He's very passionate and energetic, which I think is evident to everyone who meets him, even for a short period of time. And I can tell you, as a resident physician, I met with him during my interview at St. Jude Children's Research Hospital when for fellowship, where he was the director of the stem cell transplant program at that time. And I very vividly can recall him taking me on a tour of his lab, and he spoke very excitedly to me about graft engineering and manipulation and his vision of the future of stem cell transplant in pediatrics. He was so inspiring and easily the most compelling faculty I met with over the course of my visit. And I know this is an impression that is shared by many in the field. So today he'll be talking to us about the future of T-cell depleted hematopoietic stem cell transplantation. And I want to thank him so much for coming. And it's quite late there, I think.
Oh, it's midnight. No problem. Okay, thank you. So thank you very much, Tanya, for this nice introduction. I hope I embarrassed you not when you interviewed there. But I also remember that, and yeah, it was a great time there. So thank you also for giving me the opportunity to think a little bit about the future. And this is my disclosure. Of course, I'm a little bit biased, maybe, as you can see, towards alpha beta depletion, but I will try to be neutral, of course. So when we look in the future or we drive forward, sometimes it's good to look a little bit in the back. And view in the back mirror how this whole thing started. And it started actually with trying in the 80s to remove tumor cells, especially neuroblastoma cells from bone marrow. And at that time, especially Pat Reynolds and Rob Seeger from Los Angeles were leading the field. They used these magnetic beads, these Dyna beads. They were as big as the T cells to remove tumor cells, but later also T cells from the graft. But this was only working for bone marrow, if at all, and was not suitable for peripheral stem cells. Then peripheral stem cells came up also in children, even in small children. We were able to collect stem cells from low body weight children, and we wanted to deplete the tumor cells in this for the autologous transplantation with the neuroblastoma patients especially. And then we had two methods. One was the negative depletion of the tumor cells, and maybe easier the positive selection of the CD34 cells. Then you do not have to know the antigens of the tumor cells because you indirectly deplete the tumor cells. That was the whole start and the intention actually to purge neuroblastoma cells. And then we got some news from a young student who published a paper and worked on some microbeads. This is shown here. he developed these microbeads. These are size of 50 nanometers, like virus particles, so you cannot see them on morphology, but they are enough to make the cells magnetic, so you can change, or you can separate the cells, either positive or negative, in a strong magnetic field. And this was this young student, Stefan Milteny. And this was his wonder magnet, very strong, as a 1.5 Tesla MRT. It's the same field strength. So if you put your Rolex in there, don't do it, it's gone. And then we were very proud to present our first data in the 80s on the conference in Mexico. And you can see this was still on bone marrow processing, purging and processing. There was no word of peripheral stem cells or anything else. So it was all bone marrow. But we already described that with this technique, we not only could purge tumor cells from bone marrow, but we could also purify CD34 stem cells. And we could also show already that these magnetic microbeads do not damage the CD34 cells. That was, of course, in the 80s, very important to see whether this would have any biological impact. Then we developed ourselves this method. This was before the Clinimax was available. You could see here we stole the perfusers from the floor. The nurses were already mad at us because we took the perfusers away, but it worked very nice. This method was a completely closed system. And we had good experience and good results to do CD34 positive selection with that approach. And then, of course, then later in '95, the Clinimax came up. And so on. And then, of course, these methods got more developed and more automated. And this was the development. We started with the CD34 positive selection. Peter Lang at that time was very leading this. And then at St. Jude, we started to do the CD319 depletion. Ray Barfield was a fellow. And also still at St. Jude, we started to try whether we could deplete alpha-beta CD19 from peripheral stem cells. Stanley Caleff, Tanya, you will know them still maybe, was also a fellow at St. Jude. And then, of course, later the prodigy came up from the Milteny company. And of course, this prodigy is really a prodigy because it does everything what you want him to do. And this in a environment where you might not need in the future all these GMP labs and specialized labs, which also helps, of course, later to maybe increase the method to other centers who do not have these specialized labs. And you can see here, I think Wolfgang has shown this yesterday, already the log depletion with the alpha-beta depletion was best compared to the previous method. CD34 was also good. The median log depletion, CD3, less. But you can also see there were some depletion which did not go well. And in a haploidentical setting, the number of T cells which remain in the graft, especially with this log depletion, are too high. And as you can see, with the TCR beta depletion was very reliable. So we had never a problem with a run which did not go well. This is with the prodigy. So the prodigy does the same. And the good news is also with the prodigy, we have a very good CD34 positive recovery. So why do we want then to perform ex vivo T cell depletion? Of course, we want to prevent graft-versus-host disease. We want to be able to omit short and long-term immune suppression. Then we want, of course, enable early post-transplant immune augmenting therapies. I think we have heard a lot about this in the previous talk today and also yesterday. This is the method. This has been also shown yesterday by Wolfgang. We use this biotinylated anti-TCR alpha-beta antibody, BMA-031. So this stands for Behringwerke, and an antibiotin antibody. So a second step that results in a high load of these magnetic beads on the cells. This is why they are retained so well in the magnet. And of course, the alpha-beta T cells are the waste, but after Matt talked, I would not describe them anymore as waste. So they are very nice products to further use them. And then, of course, we have the NK cells and the gamma delta T cells in the graft. And I think this has been now talked a lot. This is and the most important is this I wanted to mention. because I cannot see this on myself on the slide. So that immunotherapy starts already at day 0. This with the infusion of these cells, and this might be the difference also to the post-Cy, which we had also with beautiful data from Ken Cook. But here we start much earlier without immunosuppression, with immunotherapy, with the infusion of these effector cells. And this is the platform, and so we have the preparative regimen, we have the alpha-beta depleted cells with the NK and gamma delta T cells, no GVH prophylaxis, and we have immune augmentation. So what could that immune augmentation be? So this is our own experience with post-transplant immunotherapy. Based on this was still with the CD3-19 depletion. At that time, we didn't have yet the alpha-beta ready. This was a phase 2 feasibility study using an antibody in children with relapsed neuroblastoma. The pediatrician, most of you are pediatricians among you, know the patients with relapsed neuroblastoma or refractory neuroblastoma. Small children are very hard to treat. And all of these children had already autologous transplantation. So we did a phase 2 study with a haploidentical transplantation after they relapsed after autologous transplantation with high number of NK cells, because these are normally small children. So you get high numbers of NK cells, of course, followed by this anti-CD2 antibody. 6 to 9 cycles every month, 1 cycle. This was especially to activate the antibody-dependent cellular cytotoxicity of the NK cells, which we infuse here. Cycle 4, we started a low-dose IL-2 treatment when we were sure there was no GDH to further boost the ADCC activity of the NK cells. This is shown. This is the results is shown here. So you can see here we did 68 patients with this protocol. The protocol is now finished. This is the 3-year overall survival with 58%. I think for neuroblastoma, this is quite promising. This is the event-free survival, 3 years still 47%. And when the patients are in remission at time of this transplant after they relapse and got another salvage therapies, they have still a 3-year event-free survival of 62%. If they still have some residual tumor, very good partial remission, we see still 53% event-free survival. If they have progressive disease, this method is not working, and I would not recommend it to do it. This is the therapy-related mortality. At day 100, we had 0, and at day after 1 year, we had 2.9%, also very low. And this is the later on the graft-versus-host disease that was also very acceptable. So I think this is one example how we can do post-transplant immune therapy on a T-cell depleted platform. At that stage, CD3, CD19 depleted. And what do we do next? I think if you can see here, these are the patients. We still see relapses later. This is where we stop the antibody, and this is a group which we also treated prior to the availability of this antibody with haploidentical transplantation, same conditioning regimen. It's a historical control group, not randomized, so CAVE. But this is our results without the antibody, and you can see there is a good difference in those patients who got the immune therapy in contrast to those who did not get the immune therapy. So my conclusion is we have to do a longer kind of immunotherapy here, most likely. So what about leukemia? We tried the same approach in acute lymphoblastic leukemia, and our immunologist, Professor Jung in Tübingen, he made an anti-CD19 antibody, a chimeric one, and introduced this mutation of 2 amino acids in the CD2 domain, which makes a very good ADCC, a much better antibody-dependent cellular cytotoxicity with NK cells. But I have to say that gamma delta T cells also have the ability to exert ADCC. So both effector cells can do it. And this antibody has no complement activation, no direct cytotoxicity. This is shown here when we incubate, when we take NK cells after transplantation, about 3 weeks after transplantation from a patient against fresh leukemic blasts. So we can see that there is some cytotoxicity, but if we add the antibody and exert this ADCC against the leukemic blast, we can boost considerably this NK activity against the leukemia. And also very interesting, this has been published by Peter Lang, that this ADCC is the stronger signal than the inhibition by the CUR, because that could also be that our CUR-HLA interaction is inhibiting the NK cells, as shown here. And if this happens, if there is an inhibition, there is no cytotoxicity, as you can see here. But if we add this antibody and activate the Fc receptor, this is the much stronger activatory signal, the most strongest probably for the NK cells, and then the cytotoxicity goes up, as you can see. So we can completely override this inhibitory signal. So the CUR constellation does not play a big role probably if we give this antibody post-transplant. So we have done this this was one case report in a patient. This patient had a refractory leukemia, had already a relapse after a standard transplantation. He was never MRD negative, as you can see here, by PCR and FACS MRD. Then we transplanted him, which we normally won't do with a second transplant when he was, while not in MRD negative remission, because we then intended to give him this antibody post-transplant. And this is what we did. He got a second haplotransplant with alpha-beta depleted stem cells, and then 2 years of this antibody. This is 1 year for every 2 weeks and 4 weeks for every 4 weeks in an outpatient setting. And then after this patient is still in remission as we speak, 6 years now, or more than 6 years in complete continuous remission. So that led us to treat 15 more patients. These patients all had a relapse after transplantation and got a second transplant, haploidentical. No TBI, of course. We use our thiotepa fludarabine melphalan scheme. And 4 patients already had a relapse after a second transplant. And for this patient, this was the 3rd transplantation. And this was followed by this 2-year antibody treatment. And this is the overall survival. And this is the event-free survival. So we can, at least half of the children, we can prevent the relapse in these patients. The transplant-related mortality was low. Only 1 out of these 15 patients had a TRM, and these are all second or third transplants, remember. And this is also our control patients where we did the same, but we didn't have that antibody yet available. So they all relapsed after without post-transplant immunotherapy. The antibody was very well tolerated, and we are planning now a prospective clinical trial. There is now an antibody against CD19 approved in the US for diffuse large B-cell lymphoma, and we hope we can use this antibody for a trial for post-transplant immunotherapy in this setting, either in MRD positive patients post-transplant or as a second or third transplantation. And these patients are at high risk of relapses, as we know. This is the probability of relapse if the patients get the antibody, so we can reduce the relapses. And this is also interesting that all these patients reconstitute their T cells again few weeks and months after we end this CD19 antibody. They recover all their B cells. So none of the patients is without B cell function at the moment. Then what about CAR T cells from the donor? This is also an interesting topic I think we have to look at. We had a patient, 17-year-old, also very severe toxicity after conventional therapy, refractory disease. Then she got autologous CD19-22 CARs, had a short response, but then progressed again. She got another boost, no effect. So multiple chemotherapies, only partial responses. And then we did a MUD transplant, not a haplo, but MUD transplant with T-cell depleted graft to omit immunosuppression. And because then we had the intention already from this donor to produce CD19-22 CAR T cells and give it to her after transplant. So she got into the transplant with residual macroscopic disease. And this is then her CAR T cells. We infused 3 million per kilogram body weight of this CD1922 CAR T cells. They showed a nice expansion. As you can see here, she only had a very mild few days erythema. Of course, once it's an erythema, you have to call it graft-versus-host disease. But sometimes it disappeared by itself. They hadn't to do anything. And this is the response. This was 4 weeks after transplant. This is a PET MRT. You can see here the lymphomas. Her bone marrow was also contaminated with blasts, and she had more of these lesions. This is 8 weeks after transplant, and then we gave her the CAR T cells. And when they expanded, then all these lesions disappeared, and she also the bone marrow went into remission. And this patient is still in remission as we speak at the moment, now more than a year after this therapy. So now to look in the future, I think that was my task. And of course, it's not so easy to look. So I brought my bowl here, and so let's have a look together. So I think this is what we have seen already. We have this platform. We can give monoclonal antibodies. I have shown you examples with CAR T cells, but we have heard we can give bites. And I think there are plans in the COG to do this. I think it's a good idea. We have CD45RA depleted memory from Brandon. We can give very specific T cells. I think also activated NK cells or gamma delta T cells, maybe soletronate activated would be something. We are also working on CAR NK or CAR gamma delta T cells because we hypothesized that this would not cause graft-versus-host disease. Also, we do not think that graft-versus-host disease is a big problem with the infusion of CAR T cells. We have experience in 2 patients with haploidentical CARs, who did not get graft-versus-host disease. Then but we also, in other approaches, we have heard also of tumor-specific T cells, WT1 or so. I think there is a lot coming up, and we can think, but also we have to think on the depletion and maybe on the preparative regimen, because I think we all agree, or most of us agree, that we want to get rid of TBI in these patients, especially in children and the smaller children. So how about the CD45RA depletion? And this is something which you have seen. Then we give the CD45RO T cells. This is very interesting. So we had a fellow in the lab, Hisayoshi Hashimoto from Japan. He worked with us for 3 years, and this was his task to see whether we can identify maybe better alloreactive T cells. So he did a mixed lymphocyte culture for 4 days from with a stimulator and donor cells and recipient cells or responder cells. And then what he did, he depleted this mixed lymphocyte culture from CD45RA. So he had only CD45RO positive T cells. And then he checked whether he could deplete these T cells which produced cytokines, which are stimulated. And then if he looked into the CD8, you can see that he could deplete some of the cytokine-producing T cells by CD45RA depletion. This is for interferon gamma T and F. And if you look at the CD4 cells, you can see that you cannot deplete CD4 cells by CD45RA depletion. You still have the same cytokine producing spectrum in these cells. So the CD4 positive cells are somehow the ones which maybe the problem maker later, and maybe the ones which cause graft-versus-host disease. So we looked further into the CD4 population on the surface of these cells and found that they express the CD276, also known as B7H3. Remember, PD1 is B7H1. Or PDL1 now is B7H1, so this is of the family of checkpoint, but we don't know the ligand for this. It's not known, but we got interested, of course, in this. And then what we did, we depleted, or what Hisayoshi did, he depleted further than the CD45RA from the CD276 positive T cells after the mixed lymphocyte culture. And then you can see, then he could deplete also in the CD4 compartment the cytokine producing cells, interferon and and tumor necrosis factor alpha. So that gave us a hint maybe that these alloreactive T cells might be in this compartment or CD276 positive. The response to viral antigens was not affected, that was maintained. And also very interesting and a hint that these are alloreactive T cells is that the response to third-party mixed lymphocyte reaction was not affected. So against the third-party stimulator, the T cells were responsive. Only against the responder cells, they were not responsive after the depletion with this approach. So we used this in a mouse model. This was a DR4 knock-in mouse, and we used a DR4 negative donor to induce graft-versus-host disease. And then we gave this negative depleted T cells, 45RA memory T cells. And you can see here, this is the bulk CD4. Memory CD4, CD45RA depleted, and then with the additionally 276 depleted memory CD4, we could abrogate the graft, improve the survival, and this is the GVH score, which we could also decrease quite significantly with this approach. So another mouse model was the same. So we induced graft-versus-host disease with CD4 cells, which were CD45RA depleted. And then we treated these mice with a CD276 antibody, which was produced by Hans-Jörg Bühring in our Immunological Institute in Tübingen. And you can see here that if we in vivo deplete by this CD276 positive T cells in these mice, the mice do not get graft-versus-host disease. But in those mice where we do not treat them and give only the CD4, CD45RA depleted, we get quite extensive graft-versus-host disease, as you can see. So that might be one hint that we can maybe further narrow down the allogenic or the alloreactive T cells by this. But how about further? What can we do further? Of course, we have to eradicate the patient's myelopoiesis to allow the immune system to engraft, and we have to tolerize the patient's T lymphocytes. I think this is the most important for the engraftment if we want to reduce the conditioning regimen. For this, we have developed a method, flow HLA method, where we can easily discern the origin of the cells. In the haploidentical setting, we have this biological marker in the setting of HLA disparity. So this is a donor A3 positive, recipient A3 negative. So you just take an antibody against HLA A3, and you can exactly tell which is donor derived. All B cells are donor derived after transplant. Here the T cells are mixed 50-50, and K cells are donor derived, and so on. This is a very easy method, very fast method in haploidentical transplantation to look at any subpopulation you are interested, and you don't need a lot of blood. This is very interesting because often we only have 10-20 microliter blood of T cells initially after transplantation, but you can still see with these small tiny amounts whether they are from the donor or from the recipient. And we have checked that, and we have seen that if we have, after conditioning, we have autologous T cells still remaining in the first days after the transplantation, as you can see here. And this is to a certain amount, these patients have a high risk of rejection because these autologous T cells will be increasing and then reject the graft. In those patients where the T cells are disappearing slowly, they have a safe engraftment. And this is shown here. The risk of engraftment is high if we have more than 60% autologous T cells within the first week after haploidentical transplantation. And with this flow cytometry, you can still see like a MRD of determination of T cells, whether they are donor recipient derived. Even if only 5 or 10 T cells per microliter, they still play a role because they will expand and they will reject. So, of course, we have heard today from the pioneering work of Ephraim Fuchs and Leo Lutznik on their approach of the post-transplant cyclophosphamide. And what they do is they tolerize alloreactive donor T cells with the post-transplant cyclophosphamide. And this is the scheme. We have seen it. Of course, this is the conditioning regimen, the T replete bone marrow or stem cells, and then cyclophosphamide to tolerate the donor T cells. So the question is, can we do the other way around for the alloreactive recipient T cells? And so we have done this. So we give an alpha-beta depleted stem cell graft. Then we don't need immunosuppression because we don't have to take care of T cells. The preparative regimen should eliminate residual recipient T cells, but if they start to expand again, and we did not eliminate them completely by our reduced conditioning regimen, the cyclophosphamide should tolerize the recipient T cells. Whether this is day 3 and 4 is the best time point, we don't know yet, but this is what we took over. But there might be a lot of room for research whether we have to give this later or earlier, depending a little bit on the flow chimerism analysis of the recipient T cells. So we had a patient, 22 years old with a combined immunodeficiency. She was very sick in a very reduced conditioning condition. He had a lot of infections. He came to us with bowel disease, lung function bad. She was traveling around the world to seek somebody to do a transplant on her. And she had no donor, only haploidentical. And we also could not imagine that she would ever tolerate a conventional transplantation. So we talked to her that we would try this combination of alpha-beta depletion and post-Cy. So we had frozen backup of every virus T cells. We had 45 or 80 depleted T cells in case she reactivates all her viruses. This was the conditioning regimen. Unfortunately, she had a fast engraftment, no graft-versus-host disease. And very important, this is the T cell reconstitution. As I expected, she had a very high increase of autologous T cells because the conditioning regimen was not very strong. But these T cells did not reject what we have seen in the other patients. So we think that they were tolerized by the post-Cy, and then the donor T cells took over, and she's now more than 5 years out and well. So we had a second patient, also very sick, a CAMU2 deficiency, immunodeficiency, only recently described. This patient was very sick for 3 years. She had warts all over her body and had all the infections you can read here. Had also decreased lung function. So we also thought this is not possible to do a transplant on her. And there have not been many patients been transplanted so far. There was only one patient in the literature, and this was a small child. So no young or adolescent has yet been transplanted. And this is the patient. You can see she was over with these warts all over her body. And then this is 5 months post-transplant, and this is now 2 years post-transplant. So most of these warts disappeared after she had a good immune system again, and she did so far well. So the question is, can we replace ATG? We had a long discussion yesterday, and I would also like to replace it because we know it has some negative influence on what we're doing. So, and maybe some of you remember our former protocol in the 90s. We didn't use ATG, we used this OKT3, the Moronumab anti-CD3 antibody, starting earlier with the conditioning and over the transplant to tolerize or to delete T cells. The nice thing was when you stop this antibody, it had a very short half-life. Then the next day, the antibody was already gone out of the system. So it was very good to manage. So no long half-life. And then we remembered that we used this anti-CD3 TCR alpha-beta antibody with our alpha-beta depletion, this BMA-031. And there was already clinical work with this antibody, which we then found in the 90s. They used this murine antibody to treat graft-versus-host disease. They have published here 7 patients and 5 responded completely. And you can see here that a nearly complete decline of circulating T cells was observed, but the T cells returned very rapidly after 1 week after the last infusion. Also very good to manage in contrast to Campath or ATG, where you have long half-life. So that gave us the idea to further work with this antibody. And there was already at the same time the company Genzyme and Behringwerke, I don't think they exist anymore. They made already a humanized antibody against this alpha-beta T-cell receptor. And this antibody induces the activation-induced cell death. This is shown here. When you have activated T cells and you engage the alpha-beta receptor with this antibody, the T cells go into apoptosis in contrast to non-resting T cells. The antibody is not affecting them. So Gregor Blank in our lab used this antibody to also see whether we can use this antibody for preventing graft-versus-host disease. And this is shown here. When he stimulates T cells with CD3, CD28, and when this is unstimulated T cells, so there is no apoptosis when he adds the antibody. When he stimulates the T cells and adds the humanized anti-TCR alpha-beta antibody, you can see here most of the cells go into apoptosis. So this is really activation-induced cell death. And the proliferation shows the same, that he can inhibit the proliferation of activated T cells with this antibody. So he also tested this in a mouse system, and he could show that this antibody abrogates aggressive graft-versus-host disease. This is shown here, especially depending on the dose and on the schedule. If he gives it as a maintenance longer, he can prevent or ameliorate quite considerably graft-versus-host disease in these mice. So this was the histology. You can see here with this preemptive doses, before we infuse the T cells, there is no T cell infiltration anymore in these organs, and this is very similar to the thymoglobulin control, which is shown here. So this antibody might also work. So the question is, can we get rid of this ATG and use this anti-TCR alpha antibody? So we are trying, of course, now to find some sponsors who would maybe help us in production of this antibody again. We are talking to some companies, but I think that would help a lot to further be able to get rid of ATG and also to decrease the conditioning regimen. So another possibility would be to increase the ATG to further or use Campath. In Germany, we do not like Campath as in the UK. I don't know in the US, but because the problem of this is that we have long half-life, and we all know that compromises the immune recovery. So increasing the ATG, you might think, oh, this is not a good idea. But there is an interesting drug out there now, which is called imlifidase. And this imlifidase is an enzyme produced by streptococci. And this imlifidase cleaves human IgG, as you can see here. And this makes this human IgG inactive, because then this antibody cannot activate complement anymore and make no ADCC anymore. And this antibody is now approved in the US at least. I think in Europe it's prior to approval as name Idefirix. This is indicated for desensitization treatment of highly sensitized kidney transplant people who have a positive cross-match to decrease their IgG antibody, of course, against the organ. And the good news is that imlifidase also cleaves and inactivates rapid ATG and Campath. And we are working on that now to see whether we could maybe increase the dose of imlifidase of ATG. This would be, for example, something, of course, this is under construction. We have more ATG or Campath, and then we inactivate it with imlifidase before we go in with the stem cells. And we hope we might get sometimes rid of the TBI also to make place in the bone marrow, as always said. And also one idea is to split the graft in two parts. One is one part, and this part of the graft is only meant to stimulate alloreactive recipient T cells, because we want them to be stimulated, because then we want to tolerize them with cyclophosphamide. And if we are worried that maybe cyclophosphamide compromises our graft, we can give the other half after cyclophosphamide, and we don't know yet whether this would have any influence But it's always good if the stem cells have not seen any chemotherapy, at least from my gut feeling, but we have to show this in the future. And then this could be also then maybe followed by CD45RA or CD276 depleted lymphocyte infusion. So, and this was the last talk, will we ever be able to use a chemotherapy-free conditioning regimen for our patients? Of course, this is our goal or our dream. And this is, again, where the CARs come into the play, and you know how the CARs are made. You take this single chain fragment from the antibody and transduce it into a T cell. And then, of course, we remembered again how we do the alpha beta depletion. And it's a very easy principle. We have this antibody against biotin. This is the single chain fragment which recognizes the biotin. And we have the biotin-related antibody. So this is the principle of alpha beta depletion. So we thought, can we do a CAR out of it? So we took this single chain fragment of this antibiotin antibody and made a CAR out of it. And this time an NK CAR with an NK cell line, because this is easier. You don't have to purify NK cells all the time. But it also works with T cells. We have done a lot of work with T cells, and the paper should soon come out. Then we have an adapter CAR. So this adapter CAR only recognizes the biotin. And whenever this recognizes the biotin, and this biotin is labeled an antibody, and the antibody binds to a target, this will kill the target. This is shown here. So this is no antibody. The NK CARs, the antibiotin CARs, because they do not recognize anything. This is the nice thing. They only get activated if they see the biotin-related anti-CD19 antibody, but it works with any other antibodies. I think we have at least tested 30 or 40 biotin-related antibodies, and it works with any biotin-related antibodies. It works also with fragments, FC fragments. So if you want a short half-life of these antibodies, you better take a biotin-related fragment, and because then you can inactivate the NK or the CAR activity. So this is something which we also think. Of course, this is still also under construction. So we make donor antibiotin in CARs. And then we can use a biotin-related anti-TCR alpha antibody to eliminate the recipient T cells. And this would have give us two birds with one stone. We would have the antibody-induced cell death with the antibody, and we would have an NK-mediated killing. So we would be maybe more able to kill these recipient T cells, which I think is very important. Or, but this is also still future, or this biotin-related anti-CD276 antibody also to eliminate alloreactive, in this setting, recipient T cells. And of course, we hope that maybe we can also get rid of the biotin-related, of the TBI and use maybe a biotin-related anti-CD3 antibody to reduce or eliminate recipients myelopoiesis. When I just added this after I heard the talk of Agnieszka that maybe biotin-related anti-107 antibody may also play a role. So this is all a little bit for the future. And of course, we also look into the possibility to use these approaches then in the organ transplant setting. We have experience and we have published this. We put together 10 patients, some of them transplanted in our institution, and out of the literature of these 10 patients, 9 are alive, which we published here, who received the organ transplant from a haploidentical donor and also haploidentical stem cell transplantation. All these patients are without immunosuppression, and this was mainly for liver and for kidney, but I think we also have to, as Alicia already pointed out, also look for bowel transplantation, because this is a major challenge in the organ transplantation. But I think slowly we might come there with our ideas if we put them all together. And with, so this is my look in the future, or my suggestion. Big philosopher Yogi Berra already taught us that it's tough to make predictions, especially about the future. So maybe we will see what's coming out of these ideas, and other ideas you all have, of course. But it's good to have these meetings and symposia to put these ideas together. I think we all learn from each other on new ideas. And this is my thanks to all my people whom I worked with, and this not, of course, these are many more, but I want to mention one guy especially, and this is Stefan Milteny, because without his invention 30 years ago, I think we would not have had this symposium. We should not forget that this changed a lot of the field in cell separation and allows us now to do a lot of this kind of new methods, and also not only the magnetic microbeads, but also all the other ideas they have with their prodigies and so on. And at the end, I think I talk for all in the name of all of us, that I want to confer special thanks to Alice and her wonderful team for organizing this beautiful symposium. I hope this is not the last one. Hopefully not. We learned so much from it. And I thank you all for participating. Thank you very much.
Thank you. That was fantastic. So in oncology pre-transplant, we often discuss sequential use of CD19 targeted therapies. So, you know, somebody who's received blinatumomab, can they receive a CD19 CAR? So in your prospective transplant study where you're using the chimeric CD19 antibody, how do you feel about pre-treatment with CD19 directed therapies prior to that?
Of course, this is easy to look. You just check whether your CD19 is still there on the cells. Interestingly, we do not see, when the patients relapse with this post-transplant CD19, we do not see CD19 negative relapses, because the immunological pressure doesn't seem to be that high. So we still have then the possibility to go with blinatumomab or with CAR T cells or whatever. But of course, we have to do now the multi-center study. We planned that in Germany with this antibody now, which is approved in the US and hopefully soon in Europe. So I think there are some maybe interesting approaches we can do. Of course, this is easier. This antibody is an outpatient infusion. It's easier to do. It's had no side effects, but we have to do the study and do this normal dose escalation study, which is now planned. Blinatumomab, of course, is also something interesting, and it's also safe to do. We have done this in a lot of post-transplant patients with relapses. We do not induce graft-versus-host disease. Also very interesting. So I think you can also start very early with blinatumomab in very high-risk patients. But of course, it's easy by flow to check just your CD19 antigen. That's much easier than in other tumors, whether it's still there.
Yeah, the other thing I'm wondering is, how did you choose the duration of that antibody treatment? Because it's limited. And how did you decide when to stop that?
That was interesting. So we did it for the first year. We did every 2 weeks, and then we wanted to stop. But then the parents they refused to stop. That was the problem, because they know this is their last chance, you know, of their child. Another relapse, is it? So they just refused to stop. So that was the compromise with the parents, to do it every 4 weeks for another year. They insisted on this, and it's hard to talk to these parents, you know, that it was not very scientific, but the patients tolerated very well. And the next study, we will study this in a really prospective study. And of course, we did it under MRD check every 3 months.
Yeah, great. Well, I don't see any additional questions in the question and answer box, so I'll transfer this over to Alice.
Thank you so much, Tanya. Thank you, and thank you, Rupert. This was a mind-blowing talk. I don't know how to define it. I know you since let's say I started to really be interested in cell depletion, and I feel very lucky that I had the chance to interact with you when I was still the resident. So really, thank you so much for what you offer us today. I am going to share the screen to conclude this other incredible second day of Symposium, and I wanted to share with you a few slides about the main messages that really we received today with the Symposium. We started really with 3 rock stars, Franco, Brandon, and Ken, Franco showed data on the update results of alpha-beta T-cell depleted. These are data in Fanconi anemia that have been published a year ago, and is the first really largest series of patients who show the 100% survival, with only 3 patients experience graft failure or poor graft function. And he reminded us, as CMV and adenovirus remain a problem in this platform, more adenovirus than CMV, as other speakers already yesterday highlighted. And he also showed the update of the 80 patients cohort that we published back in 2017, that now account up to 150, which really confirm the great GVHD-free, relapse-free survival at 10 years in this case, with still an advantage of using the TBI in the conditional regimen, but very importantly in this updated cohort, this advantage is true for ALL, but not anymore for AML. So a practical message to really bring us in our clinic. Brandon from St. Jude gave us an overview of their experience with the platform of T-cell depleted haplotransplant, and how the utilization of CD45 naive depleted DLI post-transplant have been utilized there, initially just on first allogeneic transplant, and now also in a new clinical trial for refractory disease. Interestingly enough, he really showed the early immune reconstitution of these patients after the DLI, with a contribution pretty early on of these cells expanded after the infusion, which contributed in a lower incidence of viral infection in comparison with the prior cohort, in which it was used the CD3 depleted strategy alone. And very interestingly, the important message that we always have in mind, really the expansion of CMV-specific CTL as a main tool to clear the infection. Also, how this outcome is improving in this nice comparison between 3 groups. As he was highlighting, yes, for sure, in the last years, we have a better supportive therapy, but definitely the success of this last approach is driven by next generation strategy of graft manipulation, which allow for less GVHD and better control of infection. A look on the future, and we are looking forward to hear at ASH some data about this combined CD45 array negative depletion and alpha beta depletion, along with the BITE. Can, in his birthday, compete with 2 strong advocates for T-cell depletion, and he showed us really an excursus of the Johns Hopkins post-transplant site evolution, with a number of clinical trials which are opening. One with NK cell after the post-transplant site to promote graft versus leukemia, and another one very interesting using multi-specific T cells, again, to promote control of post-transplant malignancies. He also mentioned the prospective trial that Adam Simons is leading within the COG, and hopefully we will be able to open soon. This trial will be a randomized trial between 10 out of 10 unrelated donor transplant and haploidentical transplant, and will also have an internal comparison between alpha beta depletion and post-transplant size, so we will gather a lot of information. A group at Johns Hopkins is also working on high-risk sarcoma, definitely a group of disease where we still have a lot to improve, but also great data in sickle cell, thalassemia, and severe aplastic anemia with the platform of post-transplant site. An exciting session in the innovative clinical trial. Agnieszka Czakowicz showed us her goal of really reaching 100% of cure for patients with Fanconi anemia, but minimizing as much as possible short and long-term toxicity. And that's really the rationale behind the clinical trial that is just opening for enrollment, and that combine alpha beta T-cell depletion with the anti-c-Kit monoclonal antibody by Jasper to substitute the TBI on the backbone of TBI 200 centigrade, flu, cyan, ATG with the alpha beta T cell depletion, and we are all excited to see what are the results that we are going to obtain. Very nice data from Joseph, produced at CHOP, showing the outcome of 23 children with bone marrow failure syndrome, but specifically with severe aplastic anemia. In this case, the donor was a non-related donor, either fully matched or mismatched for one or two antigens. They use a tailored conditioning regimen based on the disease, and they obtain really great results, even when compared with matched sibling donor. You can see here, neutrophil platelet engraftment was faster than with unmanipulated matched sibling donor transplant. And then the provocative question from Chris Borak, but also Mike Pulsifer and the PBMTC clinical trial going on. Do we really need a TBI for treating ALL, and specifically in haplotransplant? They are investigating if there is a real answer to this question, evaluating two different conditioning regimens, one containing a full myeloablative dose of TBI and the other one reduced intensity with just flu, thio, and MEL. And this preliminary analysis showed, interestingly, no difference in transplant-related mortality in these two groups, but not even difference in terms of survival by NGS MRD status. So Chris' really provocative answer in this case was, is this the power of the graft versus leukemia provided by haplo, which is making up for the absence of the TBI in the reduced intensity conditioning regimen? And another provocative question from the UCSF group is actually TBI not helping the graft versus leukemia because it is more lymphodepleting post-transplant, and so is compromising the immune reconstitution. Curious to see an update of this data. Matt showed the preclinical results of our collaboration in manufacturing CAR T cells from the leftover alpha beta T cells after the manufacturing process that have been shown at the dose of 5 million to completely eliminate the disease in an animal model, and the possibility of hopefully starting soon a clinical trial using these CAR T cells in patients with ALL, and potentially integrating an elegant mechanism of suicide gene, not using a suicide gene itself, but using a mechanism that metabolically can eliminate the T cells themselves. And then really, I couldn't think to anything better than the talk that Rupert just gave to conclude this 2 days symposium. Rupert elegantly, as usual, showed us his vision for the future and tried to put together, I would say, what are the wishes that we all express in these 2 days. Ideally, we want to have a reduced intensity conditioning regimen, definitely without TBI, but hopefully in the future without even chemotherapy. And so the importance of developing monoclonal antibodies that can clear the recipient immune system before the transplant and can boost the immune system post-transplant. Different strategies of graft manipulation that can accomplish two main goals, reducing GvHD, but promoting a good immune reconstitution, use of CAR T cells with either alpha beta, gamma delta, or NK cell platform. And moving forward, really an intriguing analysis of what are the T cells which are responsible of the GvHD, even amongst the naive subset. Is there a more sophisticated depletion that we can utilize to reduce the risk of GvHD and maintain the antigen-specific activity? And can we use a more precise way to evaluate the chimerism through, again, monoclonal antibody and flow cytometry to detect the early sign of graft rejection? Really impressive, this data that show that the presence of over 60% of autologous T cells the first week after the transplant are predictive of a high risk of primary graft failure. And then the data that really combine the two main platforms of haplotransplant that currently ongoing in the world, alpha-beta depletion and post-transplant side. In this case, post-transplant side to substitute the conditioning regimen and tolerize the donor, the recipient T cells. This can be a way to really eliminate the need of ATG, for example. But another strategy can be this human monoclonal antibody against the alpha-beta TCR receptor, which could be a great alternative to the long life of both ATG and CAMPAS. And lastly, you know, I would say a dream list of what we don't want and what we would like that Rupert is working on: eliminating the ATG, eliminating the TBI, potentially not using the cyclophosphamide if we are able to use a biotinylated antibody or monoclonal antibody that can really completely abrogate the T cells, abolish the T cells that are residual in the T cells, and then having the possibility of having an engraftment without all these side effects. And now for me is really the time of concluding, and I have 3 slides to thank all the people who really made it possible. I will need many more, but it's 1:03, so I'm not gonna spend too much time. First, I need to really thank who programmatically is rendering possible that at Stanford we are doing what we are doing, and that I can produce the data that I showed you today. The Center for Definitive and Curative Medicine and all the infrastructure that Maria Grazia especially put in place since she came at Stanford, with the GMP facility, the clinical trial program, and the CDCM structure overall, which allow us to really have the possibility of developing a translational research and to bring it to the bedside product that we develop into the bench. The BUS Center and the Division of Hematology, Oncology, Stem Cell Transplantation and Regenerative Medicine, and Tanya Gruber as our chief, who is incredibly supportive of the stem cell transplant section and our need. Mary Leonard, our very supportive department chair, the leadership of the hospital and the School of Medicine. And thanks again, Paul King and Dean Minor for the very kind introduction yesterday, and the LPC Foundation with Cynthia Brand and Payal Shah, who are generously really supporting us, helping fundraising for philanthropic funding that give us the freedom of running research while we are getting grants. So Rupert thanked Stefan Milteni. I needed to really go alongside with him in thanking him. When I called him and I told him that I had this idea of organizing this meeting, he immediately said enthusiastically, yes, I think it's a great idea. Milteni is going to support you. And that wouldn't be possible really without the support of the company. And a special thank also to Michaela and Nicolas, who have been involved in all the details of the organization. And lastly, but I would say most importantly, the people who really made it possible: Angelica Martinez, Elizabeth Alarcon, Jennifer Corey, and Maisy Mayo, who took care of all the organization with the speaker, moderators, the agenda, and made sure that everything was ready to go. I have to really thank Priscilla Slepichka from my lab, because besides being an excellent postdoc, she's the one who designed the logo for this symposium, who made the background and everything which graphically you can think about, being always excited, engaged at my request. The Stanford video team, especially Winnie and Ranian, for making these 2 days really going smoothly and very effective. The marketing office, and of course, all the 16 speakers and the moderators, which made this event much better than my expectation. I am humbled that I had this opportunity. And yes, Rupert, this is something that we are going to organize again. It has been too fun and too exciting, so definitely we will make it happen again, and I really hope the next time can be in person so that we can meet all together and also have some fun after the talk. I hope you all really enjoyed the meeting as I did, and I wish you a great rest of your day. Thank you again for following us.